Welcome to this lecture on cell and molecular biology. Today, we are going to dive into one of the most fundamental processes of life, transcription. We will explore the intricate machinery and the step-by-step mechanism that allows a cell to read its genetic blueprint. By the end of this session, you will understand exactly how DNA is copied into RNA, why this process is so critical for every living organism, and how we can even calculate the speed of this biological machinery. Let us get started. To truly understand transcription, let us start with a simple analogy. Imagine you have a massive, ancient, and incredibly valuable cookbook stored in a secure, locked library. You want to bake a specific cake, but you are strictly forbidden from taking the heavy cookbook out of the library and into the messy kitchen. What do? You take a blank piece of paper and carefully copy down just that one specific recipe. In a living cell, your DNA is that valuable master cookbook, and the nucleus is the secure library. The temporary, disposable copy you make is the messenger RNA. Transcription is simply the biological process of copying that specific recipe from the master DNA book into a usable RNA format. Now that we have our analogy, let us look at the formal biological definition. Transcription is defined as the highly regulated process by which an enzyme synthesizes a single-stranded RNA molecule using a specific segment of double-stranded DNA as a template. This is not just a random copying event. It is the crucial first step in expressing a gene. By creating this RNA copy, the cell ensures that the original genetic blueprint remains completely safe and untouched inside the nucleus, while the rest of the cell gets the exact instructions it needs to build essential proteins. Understanding what transcription is leads us to ask, what actually performs this copying? The core of the transcription machinery is a powerful enzyme called RNA polymerase. You can think of it as the molecular photocopier. However, RNA polymerase cannot just start copying anywhere. In prokaryotes, for example, it must bind to a very specific starting sequence on the DNA known as a promoter. To find and bind to this promoter accurately, the RNA polymerase requires the help of a specialized accessory protein called a sigma factor. Together, they form the complete machinery, ready to begin the transcription process. With the machinery assembled at the promoter, the actual mechanism unfolds in three distinct sequential stages. The very first stage is called initiation. During initiation, the RNA polymerase and its associated factors go to work on the DNA double helix. They physically unwind a small portion of the DNA strands to expose the internal genetic bases. This unwinding creates a localized open area that scientists call a transcription bubble. This bubble is absolutely essential because the enzyme needs direct access to the unpaired DNA bases to read the genetic code and start building the RNA copy. Once the transcription bubble is formed, we move into the second stage, known as elongation. This is where the actual copying happens. The RNA polymerase travels along the template DNA strand, reading it strictly in the 3' to 5' direction. As it reads the template, it simultaneously builds the new RNA molecule in the opposite 5' to 3' direction. It does this by continuously bringing in and attaching complementary RNA nucleotides, linking them together one by one to form a growing single-stranded messenger RNA chain. After the entire gene has been copied during elongation, the process must eventually come to an end. This brings us to the third and final stage, termination. The RNA polymerase does not just guess when to stop. It continues until it reaches a very specific stop signal encoded directly in the DNA sequence. Upon hitting this signal, the enzyme halts. The newly formed RNA transcript is released and detaches from the DNA template. Finally, the entire transcription machinery disassembles, leaving the DNA double helix to zip back up, completely unharmed and ready for the next time it needs to be read. Before we look at the mathematics of this process, we must understand why transcription is so vital. Transcription is the fundamental control point for all cellular activity, development, and identity. Every single cell in your body contains the exact same DNA, yet a beating heart cell acts completely differently from a thinking brain cell. This incredible difference exists solely because different cells transcribe different sets of genes at different times. It acts as the ultimate switchboard for life. Furthermore, many severe diseases, including various forms of cancer, are caused by errors in transcription, making this machinery a critical target for life-saving genetic therapies. Now that we understand the biological importance, let us look at how efficiently this machinery operates. While transcription is a complex biological process, we can actually understand its speed using a simple kinematics formula, very similar to calculating travel time in physics. The formula is T equals N divided by V. Here, T represents the total time required to transcribe the gene, usually measured in seconds. N represents the total number of nucleotides, or the physical length of the gene being transcribed. Finally, V represents the velocity or rate of the RNA polymerase enzyme. Conceptually, this tells us that the time taken is directly proportional to the gene's length and inversely proportional to the enzyme's speed. If the gene is longer, it takes more time. If the polymerase moves faster, the time decreases. Let us apply this formula to a real-world scenario. Imagine a typical bacterial gene coding for the enzyme beta-galactosidase, which is crucial for digesting lactose. We are given that the total length of this gene, N, is 3,000 nucleotides. We are also told that the bacterial RNA polymerase synthesizes RNA at a constant velocity, V, of 50 nucleotides per second. We need to find the total time, T. We choose our kinematics formula, T equals N divided by V, because it perfectly relates distance, speed, and time for this molecular journey. Substituting our known values, we divide 3,000 by 50. The nucleotide units cancel out, leaving us with exactly 60 seconds. Physically, this means it takes just 1 minute for the bacterial cell to completely copy this entire gene, highlighting the incredible efficiency of molecular enzymes. Having seen how fast the machinery works, let us zoom in to a microscopic level to understand exactly how the genetic code is read. Directionality is a fundamental rule in molecular biology. The RNA polymerase enzyme has a strict directional preference. It always reads the template DNA strand in the 3' to 5' direction. Because nucleic acids must align in opposite directions to pair up, the enzyme synthesizes the new RNA molecule in the antiparallel 5' to 3' direction. The machinery meticulously processes the template, one single base at a time, ensuring no information is lost. As the polymerase moves along the template, it must choose the correct building blocks. It does this using strict complementary base pairing rules. However, RNA is slightly different from DNA. When the enzyme sees an adenine on the DNA template, it pairs it with a uracil in the RNA, because RNA lacks thymine. If it sees a thymine on the DNA, it pairs it with an adenine. Cytosine on the DNA always pairs with guanine in the RNA, and guanine on the DNA always pairs with cytosine. These rules guarantee that the newly formed RNA is a perfect complementary copy of the genetic code. Let us put these base pairing rules into practice with another example. Suppose we isolate a short segment of a DNA template strand during the elongation phase. The sequence is given as 3' TAC GGA TCA 5'. We are asked to determine the exact sequence of the newly transcribed messenger RNA. We know the enzyme reads from 3' to 5', so we go left to right. Applying our rules, T becomes A becomes U, because this is RNA, C becomes G. Continuing this pattern, G becomes C, G becomes C, A becomes U, T becomes A, C becomes G, and finally, A becomes U. The resulting RNA sequence is synthesized in the 5' to 3' direction. This perfectly anti-parallel copy now carries the correct genetic information. Now that we have calculated the speed and derived the sequence, let us look at a visual summary of this entire mechanism. Here you are seeing a schematic of the transcription bubble. Notice the large oval structure. That is the RNA polymerase enzyme, enclosing the unwound DNA. The bottom black line is the template strand, oriented from 3' to 5'. You can see the red messenger RNA strand being actively synthesized in the 5' to 3' direction, perfectly paired with the template bases inside the bubble. The top strand is the coding strand, which is pushed out of the way. The arrow indicates the machinery moving steadily to the right, reading the code and building the transcript simultaneously. This diagram beautifully captures the elongation phase in action. We have covered a lot of ground today, so let us review the key takeaways. First, transcription is the essential biological process of copying the master DNA code into a temporary messenger RNA format. Second, this entire mechanism is driven by the RNA polymerase enzyme, which meticulously executes the stages of initiation, elongation, and termination. Third, we learned that the machinery strictly reads the template in the 3' to 5' direction, using specific base pairing rules where adenine pairs with uracil. Finally, we saw that this biological process can be quantified, allowing us to calculate transcription time based on gene length and enzyme speed. You now have a solid understanding of how genes are expressed. Excellent work today!